So today, indeed, I'm going to talk about a novel treatment option for USH2A-associated retinal degeneration. And well, I think this slide is rather redundant after already a very nice day of yesterday. One of the things that I would like to stress here on this slide is that mutations causing Usher syndrome are caused by a so-called loss of function disease mechanism, which is important for future treatment option development. Hearing impairment in Usher syndrome is often compensated by providing patients with either cochlear implants or hearing aids. However, for the loss of vision caused by retinitis pigmentosa, currently no treatment options are available. So looking at the genetics and causes of Usher syndrome, there's one important cause, and that are mutations in the USH2A gene. So far, we know approximately 600 individuals in the mutations that are present in this gene that can either result in Usher syndrome in approximately 170,000 individuals worldwide, or can result in non-syndromic form of retinitis pigmentosa in approximately 250,000 people worldwide. So that's quite an amount of patients that lose their vision due to mutation in this single gene. So looking at the progression of the disease, as said, Usher syndrome patients are born congenitally hearing impaired, and this hearing impairment can, in case of Usher syndrome type 2A, partially rescued by providing them with hearing aids. And at an early adolescence, the first symptoms of RP become apparent, and that are often seen in the form of night blindness. And this night blindness progresses into a form of legal blindness at an age of approximately 50 to 60 years, leaving in between a window of opportunity in which we can therapeutically intervene in order to prevent or to delay the symptoms leading to this legal blindness. So when talking about therapeutic solutions, two things need to be taken into account. First of all, you would need a functional model to assess the therapeutic strategy. That's the second thing that you will that you are going to develop. So as a functional model, we use zebrafish. And the reason for that is we are talking about vision loss caused by mutations in USH2A gene, and mouse models that are present that lack the USH2A gene or have the USH2A gene mutated only show very mild retinal dysfunction at a very late age in life. So we were looking for an alternative model, and we used zebrafish for that. USH2A is present in the zebrafish genome as a single copy, and looking at the gene and protein structure between human and fish, we see that both of these structures are highly conserved. And by adopting the programmable nuclease CRISPR-Cas9, we introduced a lesion of NAT4 base pair in the exon 13 of the USH2A gene, resulting in a premature stop and a non-functional protein. After recording ERGs already at 5 days post-fertilization, so early larvae of fish, we saw that B-wave amplitudes in the ERG were significantly decreased, indicating that these fish already are vision impaired at a very early stage, so making them particularly suitable to evaluate therapeutic strategies. So when talking about options for treatment, as said, mutations act via loss of function mechanism. So gene augmentation could be a perfect alternative. However, the size of the coding sequence of the USH2A gene, as well as for several other Usher syndrome associated genes, is tremendously large and by far exceeds the currently available capacity of the viral vehicles for gene delivery. Also, for several genes, including USH2A, multiple isoforms are present, so you need to determine which one is most important to augment. And also, expression levels are often critical and occasionally difficult to control using gene augmentation. Gene correction could be an alternative. However, there's always a risk of off-target editing and introducing permanent lesions there. And these treatments are most of the time mutation specific. And as said, already more than 600 individual mutations in the USH2A gene alone have been identified, so making this quite a challenge. And also efficiency to date is still not very high and needs improvement. And finally, also immune reactivity against endonucleases used in these gene correction strategies is also an issue. Mechanism-based treatments could also be an alternative. However, the exact biological mechanism underlying disease is often not known, and this is also the case for USH2A-associated disease. So ideally, you would like to interfere at the level of RNA, so thereby not altering the protein isoform landscape within the organs of interest and not altering gene expression levels. So the solution we chose for that was splice modulation, using antisense oligonucleotides. So antisense oligonucleotides can bind to pre-messenger RNA and prevent the binding of splice factors, inducing the skipping of exons during the process of pre-mRNA splicing. And these AONs, or ASOs, as they are abbreviated, can be used in two flavors mainly in splice correction. First of all, you can correct aberrantly spliced exons or pseudoexons. And we have shown that for a frequent mutation in USH2A, inducing the introduction of a pseudoexon 40. And we did that together in collaboration with a group of Anne-Françoise Roux. And also Jen Lenz has shown this, and Gwen Gilliok will probably elaborate on this a bit more later this session to correct the splicing of USH1C gene. You can also use splice switching antisense oligonucleotides to induce the skipping of so-called in-frame exons that contain mutations. And this is where this talk is going to be about today. So I said 600 individual mutations that are all rare have been identified in the USH2A gene. However, 2 founder mutations have been identified, and that are both present in exon 13 of USH2A. So in general, pre-mRNA is being spliced into a messenger RNA that is translated into a protein with a complete function. And in case of USH2A mutations, in this case, the 2 founder loss of function mutations, proteins are not formed or are malformed and lack total function. And by inducing this in-frame exon 13, the frame is restored, resulting in a slightly shortened protein with hopefully residual function. So that's the basic principle underlying exon skipping here. So exon 13 encodes part of the stretch of EGF-LAM domains that is present at the N-terminal region of the Usherin protein. And by skipping exon 13, this would induce the loss of EGF-LAM domains 5, 6, and 7, and the simultaneous fusion of EGF-LAM domains 4 and 8 into a functionally similar EGF-like domain. So how to assess then the effect of this skipping on the level of visual function? Well, as said and shown, we have a zebrafish model to assess this, and that's what we did. So we asked ourselves 2 questions. So first of all, can we, in the zebrafish model, induce the skipping of this exon 13? And we did that by a trial and error method for testing antisense oligonucleotides on exon skipping potential, and then selected the best performing AONs to do this. And using that combination of AONs, look at if this protein is being produced, and if so, is it present at the proper subcellular location where we would expect it, and does it indeed lead to improvement of visual function by means of ERG measurements. So we indeed found a combination of 2 AONs, AON 1 and 2, named over here, in which that we applied to our zebrafish model. And as you can see here, we were able to induce the skipping of exon, specifically exon 13, and that seemed quite efficient. However, when we quantified this, we noticed that only 22% of transcript indeed lacked exon 13, whereas only 5% of transcripts still contained exon 13. So approximately 70% of transcripts were completely lost, so degraded or at least not present anymore. So it seems highly efficient only, but if you look more closely, only 22% of transcripts were the ones that we hoped for. So is that enough? We also looked into the retina of these fish and used an antibody directed against the C-terminal part of the usherin protein. And what we noticed is that indeed a partial amount of usherin lacking exon 13 was produced. Now we quantified that using immunofluorescence quantification, and we indeed noticed that there was a significant increase, but that was approximately 20% of protein of the normal wild-type levels that was being produced. But it's present at the proper and expected subcellular location at the periciliary membrane. So we recorded ERGs in these fish, and what we noticed is that this 20% of usherin protein lacking exon 13 is apparently enough to completely restore visual function. Here you can see the representative ERG traces in blue and in red. These are the mutants and non-targeting antisense oligonucleotide control-treated mutants, whereas in the black line is the wild-type larvae, and in green are the AON-treated and exon 13 skipped larvae or mutants, and you see that these perfectly align. So we concluded from that is that indeed, if we mutate exon 13, we do see in these zebrafish larvae that no protein is being produced, and these fish show an early retinal dysfunction recorded by defective ERGs. And skipping of this mutated exon results in a partial return of usherin expression with the correct subcellular distribution, and that this 20% is enough to restore ERG traces in these larvae. So we concluded from that exon 13 skipping is indeed a promising therapeutic approach for the future treatment of these patients having mutations in this exon 13. So we moved on and translated these findings into use in man. And we performed an oligo walk, designing antisense oligonucleotides that were completely phosphorothioated with 2,O-methoxyethyl modifications. And we transfected these AONs in cultured retinoblastoma Weyeri cells that endogenously express USH2A. And on the left side, you see here in the untreated, sample, and you can see here that usherin full length is being produced, but also two aberrant fragments that endogenously are being produced, and that are transcripts lacking exon 12 and 13, and also transcripts lacking exon 12, 13, and 14. So we designed 14 different AONs, and we selected this one as our lead molecule, which was named QR421A after that. So no wild-type and full-length USH2A product could be detected anymore, whereas we observed that exon 13 is skipped quite efficiently. And we continue with this molecule. So we generated iPSC-derived photoreceptor precursor cells obtained from a patient that was homozygous for the 2299-DEL-G mutations in exon 13. And what we noticed is that we do see a dose-dependent increase in exon 13 skipping properties that are go up to approximately 60%. However, if we would translate this into a use in vivo in patients, we obviously are not going to transfect the retina with this, but it will be a naked delivery into the vitreous. So what we did, we also applied these AONs into a genotic delivery, so adding these AONs naked to the medium of cultured cells. And we also noticed a dose-dependent increase in exon skipping properties, although it's obviously lower than what is being seen after transfection. So the next step was to inject a mouse surrogate molecule of QR421A into the vitreous of mouse model. These are wild-type mouse models. And first of all, in the left upper corner, we looked at whether or not these AONs were able to diffuse to all different layers of the retina. And in red, although it's quite difficult to see in this picture, we notice that indeed, upon a single intravitreal delivery of this molecule, these AONs were distributed all the way up to the RPE and were present there for up to 259 days. And that's where we stopped looking. Also, this molecule was able to induce specifically exon 13 skipping. And by applying different doses, we observed that after 7 days, when we harvested the eyes of these mice, we do see that you get a plateau at approximately a dose of 30 micrograms of injection. And we use, therefore, that dose to follow up a time response course after a single delivery of 30 micrograms of this molecule. And what you see is that you get an increase up to approximately 40% of exon skipping, but that this exon skipping properties last for up to 200 days without really wearing off. So meaning that these molecules, when they enter the cells, are stable for quite some time there. So after some safety parameters that we have checked, phase 1/2 clinical trial was initiated by Procure Therapeutics, with whom we obviously collaborate on this. And the first patients were dosed in March 2019 in a STELLAR trial. So this STELLAR trial is actually the very first clinical trial for treatment of USH2A-associated retinal dysfunction. And this first-in-man study was mainly based on proof of concept obtained solely from a zebrafish, which is also quite unique to our knowledge. And just recently, positive intermediate results were reported, and describing that this molecule is well tolerated in all patients with no serious adverse events reported and also no inflammation seen. And that this molecule demonstrated benefits in multiple parameters of vision that were assessed, including BCVA, static parametry, and also retinal imaging by means of OCT. And finally, and that's even most important, I think, and that was also not completely what we expected on beforehand, is that this molecule, treatment with this molecule benefits all treated individuals, including the advanced staged individuals that were included in the study. And these are some results that I would like to end up with. First of all, in the left lower corner, this is the best corrected visual acuity that was recorded, and in which we observed an improvement of 9 letters in the treated eye as compared to the untreated eye. And this was mainly, that will be used as an endpoint in the phase 2/3 trial in an advanced stage population. And for the early moderate population, another parameter will be used as a readout, and that will be static perimetry, in which you can record retinal sensitivity. And also upon single delivery, you do see an increase there of the amount of foci that are higher than 7 dB. And as you can also see here, after approximately in between 12 and 20 weeks of delivery, you see that the effect wears off, which would probably mean that you would need to re-administer the molecule there again. But that is part of the next study. And as said, if phase 2/3 trials are being prepared currently, hopefully to commence before the end of the year, using an advanced population, and that will be the Sirius trial, or a second phase 2/3 trial using early moderate population, and that will be the Celeste trial. And so hopefully these results will keep up, and can inform you with some very encouraging results in hopefully the next session of this wonderful meeting. And with that, I would like to thank all of you for your attention, my team for doing all the great work, and also the people at Procure, that also work together with us in a very neat way to get all these things done, and also the funding bodies that also made this possible. Thank you very much.
Thanks, Erwin, for this nice presentation and this exciting success of this ASO therapy for vision in USH2 patients. We have time for 2 questions. We have one from the speakers. Ove Wolfram and Gwenaël has exactly the same question, asking if naturally, in natural conditions, you may have alternative splicing of exon 13.
Yeah, that's occasionally seen. Well, we do not see that in, or we have not seen that in RNA derived from human donor retina, and also not seen in our hands, at least in PPCs, so patient-derived photoreceptor precursor cells. We do see this to happen in patient-derived fibroblasts. But probably, and that's what we have also noticed before in different cell cultures, we do see that splicing differences occur between different cell types and different organs. So I don't know how much of natural exon 13 skipping occurs within the retina, but so far, what we have seen, at least in our hands, this is not a very common thing to happen.
Yeah, I have a short question and then another from Serge. Based on the results of this first clinical trial, there is a need of repeated injections or administrations. If you project in a long term, in terms of like 30 years, how much or how many repeated injections you think that might be needed?
Right, that's a very intriguing question that I cannot completely answer. I can only make a guess there. So far, what we have seen is that the molecule is quite stable. So that's one. But second of all, is you need a certain dose that is present in the appropriate cells in order to be efficient. And what you see is that indeed after, in between, let's say approximately 20 weeks, the effects there wears off a bit. So and there a re-administration is needed. So what we so far anticipate is that we would need re-administration 2 to 3 times a year. That's what we so far think.
Okay, I think short question by Serge, and then I will step in. Do you have an effect on the contralateral eye when you do an injection in one eye?
No, we have not seen that.
Aziz, I have a short question.
Yeah, Olivier, go ahead.
Yes. Okay, thank you very much. You said that you see some benefits in all the patients, including the advanced stage of the disease. What do you mean by advanced stage of the disease? And they still have some photoreceptors remaining, or you regrow the other segment? Then
that's a good point. Well, of course they have photoreceptor. This treatment depends on the presence of photoreceptor cells.
Okay.
And what we do see is that. And that was in the beginning also not completely what we expected. We do see improvement, slight improvement of visual cues after treatment. And that is probably due to the fact that at the edge of the photoreceptor, or at the edge of the visual field that is still present, photoreceptor cells are not functional anymore, but also not yet degenerated.
Okay.
So what we noticed there, also by OCT and other imaging techniques, we do see that the cell body is still there, but the outer segments are crumbled or not present anymore.
Okay.
And it seems that we can revive these cells when they are still there, but not yet degenerated. So that improves vision parameters, or these certain vision parameters a bit, but it won't go any further with this treatment.
Maybe it's like a teaser for my talk. Maybe you should try on retinal organoids, then before they get some other segment to see what's going on now.
Yeah, definitely.
Thanks, Erwin. So we move on.
Thank you very much.